Good evening. Welcome to the 619th Biweekly Assembly of the New Asia College of COHK for the 2025 to 2026 academic year. We will now sing the college anthem to kickstart the assembly. Everyone please stand and join in singing.
山林野，海深深，地国后，天高地，人之存心之力。广大出体，有着天生珍重珍重，这是我信仰精神。珍重珍重，这是我信仰精神。时光里，上下时光不要紧守。我现在进来过往里面，你是我生命之神，东海西海，深人，珍重珍重，这是我信仰精神。珍重珍重，这是我信仰精神。守护我义务都要了，不知情，万民中流浪民。我尽我的心，坚信我的心，芬芳我的心，全心当自然之条，成其中，坚强前行，珍重珍重，这是我信仰精神。珍重珍重，这是我信仰精神。
Please be seated. Students, please be reminded that you must attend the biweekly assemblies or other events which you have registered. All students must register for and attend 3 events, at least 1 assembly for each term of the 2025 to 2026 academic year. Should students fail to register for assemblies or have any inquiries on the attendance, please contact the Dean of Students' Office. In order to ensure your attendance is successfully recorded, students can check your attendance record at My Event on the registration and attendance system after 4 p.m. on the 3rd working day after the biweekly assembly. This assembly is jointly organized by the Yen Guoyong Lecture in Life Sciences. Today, we are honored to have Professor Christian R. Voolstra, Professor of Genetics of Adaptation in Aquatic Systems from the University of Constance, to share the topic of the future of reefs and the reefs of the future. We are also pleased to have Professor Lam Hon Ming, School of Life Sciences of COHK, to introduce the speaker. Professor Lam, please.
Professor Voolstra, honorable guests, colleagues, and students, a warm welcome to the 10th Yen Guoyong Lectures in Life Sciences. Since 2015, New Asia College and the School of Life Sciences have co-organized this lecture series, made possible by the general support of the Dr. Yen Guoyong's family. Each year, we have the privilege of inviting world-renowned scholars to share their insight and deepen our appreciation of the life sciences. This evening holds special significance as we host the first lecture of this series in 2026. It is my great pleasure to introduce our distinguished speaker, Professor Christian Voolstra. Professor Voolstra holds the chair of genetics of adaptation in aquatic systems at the University of Constance. As a coral reef scientist, he pioneered the concept of the coral meta-organism, revealing that corals are not solitary entities, but complex partnerships formed with algae and bacteria. It is this microscopic community that holds the key to how corals adapt to environmental stress. Christian has developed the tools to study these dynamics, integrating ecological, molecular, and microbial approaches. What truly sets him apart is his commitment to open science and practical solutions. He has developed resources such as Sympato and the CBASS system, a mobile coral fitness test designed to identify heat-tolerant corals for climate-smart restoration. He currently serves as president of the International Coral Reef Society, is scientific co-director of the Terra Coral Expedition, and has received the ICRS Mid-Career Scientist Award. With over 300 publications, his impact on the field is profound. Tonight, he offers a realistic and science-based outlook on the future of our reefs, acknowledging the risks while highlighting clear opportunities for action. Please join me in giving a warm welcome to Professor Christian Voolstra for his talk, The Future of Reefs and the Reefs of the Future. Professor Voolstra, please.
Okay, good evening, everyone. This is a really big audience. I'm excited. Thank you so much for having me and for inviting me. It's really an immense honor. And I hope, or I'll try to make it worth your while. So, who has dived in coral reefs? Okay, some people, good. This is sometimes how reefs look like at 5 a.m. in the morning when you get up on the boat. There's this tranquility and the water is like glass, and then you can really see the life below the water surface, which is really the moments I enjoy in life. But before I start with a lot of exciting science, I briefly want to introduce myself and where I'm coming from. This might be interesting for students, right? Because when I was a student, I was always asking myself, how do you become a professor? And every professor I asked gave me a different answer. And this is pretty much the answer I give you. You have to kind of forge your own path. There is not a list of things you have to do, but I think you really have to love what you do, and you have to do what you love. What you can see here is that I actually started out in fruit fly research, which I did my diploma at the University of Cologne at the Institute of Genetics, which is actually a pretty famous institute for genetics. I pretty quickly figured out that microscopy is not my thing, so I switched to mice, and then I studied evolutionary genomics in wild mice, following the path of adaptation, because mice are commensal with humans. I still didn't kind of find my thing that completely captures my heart. So as a postdoc, I completely switched gears. And I studied coral reefs in Mexico. Only then to go to the Red Sea, where I started as an assistant professor in the King Abdullah University of Science and Technology. So what I want to tell you is that at no point in my life could I have basically predicted how this is going, but at each stage, it was really helpful what I learned, and I could always piece the things together. Like, it was really good to start off in model systems, because when I started in coral biology, the molecular biology or genetics was just taking off. So I had an immense advantage over that. And it's also to tell you that be calm and do research, right? It only makes sense looking back the career path. Like, now I can explain you how this all helps me. But as I said, at each point, I basically followed my heart and what I wanted to do with my life and in terms of research. So, of course, now, ironically, I ended up at one of the biggest lakes in Europe, freshwater systems, studying coral biology, and here we meet. So life is long and interesting. Okay, but back to the coral reefs. This is a typical coral reef. If you're lucky, you get to see it like this. And coral reefs are basically pretty important ecologically and economically. Charles Darwin was actually already interested in corals, and he coined it the oasis in the desert of the ocean. So why is that so? Because corals are usually very successful when no other organism is successful. So they thrive in very oligotrophic, so nutrient devoid conditions. They are very important ecologically in terms of biodiversity. They were also called the rainforests of the sea. So 30% of the described marine biodiversity basically depends on coral reefs. And economically, the most recent estimates is actually that 1 billion people directly depend on ecosystem services provided by coral reefs. But unfortunately, they are globally threatened. And you probably saw a picture like this or similar. These corals turn white, which is what we call coral bleaching. It's important to acknowledge that these corals in the bleached state are not dead yet. They can actually recover from this state. We get back a little bit later to that. But this is how coral bleaching looks like at a reef scale. And can you see the pointer? Yes. So what you see here is a world map. This is actually part of the Tara Pacific expedition, which I had the privilege to join. So you can see this is the Pacific Ocean, and the yellow circle is where that reef is. So it's in the absolute middle of nowhere. It took 6 weeks on a boat going there, with basically the blue ocean on the bottom and the blue sky on the top. There was no land. This was my first dive on this expedition. I was super excited to go there. It's literally the reef that is the furthest away from any direct human impact. And you can imagine how I felt diving underwater there. Was basically for me the reminder that these reefs are nowhere protected in this world. So we have to better understand them. Unfortunately, this is not an isolated incident. Basically, when I was born, and well, probably many of this room were not born yet, but mass coral bleaching is a very recent phenomena. So the first mass coral bleaching, like just on a local event, was actually described in the mid-80s, and only since the turn of the century in 1998, we have the so-called global coral bleaching events. You can see that they basically increase in succession over the time, and also increased in severity. So ironically, we just published this paper with 150 authors, which describe this third global coral bleaching event. And by the time, we haven't even published the third global coral bleaching event, and the fourth event already unfolded. So I just left this here because this is a slide that I used some 2 or 3 years ago, where I just put a question mark. Who knows what will happen in 2024? And this actually, the fourth global coral bleaching event is still ongoing. But the scale is crazy. So think about that this would happen to the rainforests above the water surface, that 80% of the rainforest basically, the trees would lose all their leaves. I mean, this would be in every news, and yet this is unfolding and we actually hear very little about it. So the fourth global coral bleaching was so severe that NOAA, which is the National Oceanic and Atmospheric Administration of the US, they had to invent a new color scale to actually display the increase in water temperature, which is this pink and purple scale here. But you can see that, so blue is no stress. Tell me if you see one blue pixel in there. Like everything was basically stressed. Okay, but what is coral bleaching? It was already well said in the introduction. But if we want to understand coral bleaching, we have to understand what a coral is. And a coral is a so-called meta-organism. That's a relatively new concept. And it basically means that none of us, no animal, no plant lives in isolation. We all team up with an array of microbes, and this array of microbes actually contributes to our biology, physiology, and also fitness. So corals, depending on how you do it, they are actually pretty smart. There's an animal, a sessile animal, that learned to live like a plant. So these coral animals, cnidarians, they host photosynthetic algal symbionts inside their tissue. And I mean, algae are plants, and what do plants do? They do photosynthesis, which means they produce a lot of sugar. So hosting these algae actually covers almost the entire dietary needs of the coral. What you can see here in large is a so-called polyp. So this is the basic building block of a coral. So coral colonies are composed of millions of these polyps, which are basically, everybody knows a jellyfish. Imagine an upside down jellyfish. Then you have a coral, and then the tentacles you basically fill with these golden dots, which are the algae that do photosynthesis. So coral bleaching then describes the loss of these algal symbionts. So this has, of course, two consequences. A, the color is lost because the algae with their photosynthetic pigments are lost. And B, the source of nutrition is also lost for these coral. More recently, we also figured out that bacteria play a pretty big role. I will touch upon this a bit later. I also think that this mechanism is reasonably well understood, so I don't want to touch too much on this. But the important point is that corals are amazingly or surprisingly temperature sensitive. And if you think about why that is so, there is very little space in the ocean where coral reefs or corals can actually grow, right? You need a sunlit, shallow environment. So by definition, this is basically the shorelines in the tropical zone. And because space is at a premium and so valuable, all these corals become competitor to each other, which means that they basically maximize their growth at the medium summer temperatures. So they become so efficient that any slight increase above this temperature is actually detrimental to them. So if you go basically to a coral reef, you can look something up which we call MMM, maximum monthly mean, which is the medium warmest temperature in the year. And if you increase the temperature only by 1 or 2 degrees Celsius, the corals start to bleach. So what we have is a situation where even under the most benign climate change scenarios, corals will bleach, right? And this is this RCP 2.6 relative concentration pathway. This is actually the best scenario which we managed to cross very successfully as humans. So we really have to do something about it. But if we talk about coral reefs and corals. So how is this connected? So corals are basically the building blocks, or how we call it in ecology, the foundation species or the foundation meta-organisms of coral reef ecosystems. They are pretty complex, but what you basically need to know for the rest of the talk is that there's 3 important players, which is the coral host animal, the algae, which we call zooxanthellae or Symbiodiniacea, or and the bacteria. And all the other players are pretty much not so well understood, but they may also play important roles. But this is also something I always say, right? It's this is not me as a scientist raising alarm. What I tell you is actually the official statement by the IPCC. So what is the IPCC, International Panel of Climate Change? What you need to know is that this is actually a consortium of scientists, stakeholders, politicians, and country governance. So they all sit together, and this is a consensus document. So what does it mean psychologically? That usually what you find in these documents is the most benign language, because everybody needs to agree to it. So even in this special report, you can read verbatim that there's a coral reef, there's a very high risk of that we lose almost the entire coral reefs at the projected 1.5 to 2 degrees warming. So in my world, then there is, of course, the Stockholm Resilience Institute, which published a report last year as a follow-up that basically said that we reached or surpassed this climate tipping point that was referenced in this IPCC report. So, of course, this is not helpful, right? Because if you say tipping point and you're a scientist, you understand that the definition is usually that this is an irreversible path. But if you publish reports like this, then what you cannot let happen is that basically people use this as an excuse to not do anything, because you basically cross the tipping point and there's no reversal of this. And I remember this report came out, and the next day I was basically doing a radio interview, and I told them that we need to use this as a basically warning sign, and that now is really the time to kick into action. And then I was asked if I'm saying that 160 climate scientists are wrong because they declared it's the tipping point. So this is the situation where you get pushed into, okay? I think it's important to acknowledge that both things can be true at the same time. It's a very dire situation, but it's not hopeless. And this is kind of what I try to convey during this talk. So 2019 came this report out. I was invited in 2018, sorry. Yes, in October 2018, the report came out. I got an email from Monaco. This is actually the Prince, His Royal Highness of Monaco. This is me. Okay, so even as a coral scientist, you have your moments of very important person publicity, but it was nice. I got an email. The Prince Albert is actually very engaged in environmental issues. He's a diver himself. So it was pretty much unbelievable. This is the who is who of coral reef biology, and me, and I was actually invited because somebody called off, but this meeting was very important, right? Sometimes it's good to pack a lot of scientists that want the same thing in one room and have a discussion. So what came out of this is this paper, which actually changed a lot of my thinking on how I deal with this issue, or how I deal with coral reefs in the real world. So, of course, so what we did is we modeled the trajectory of bleaching going forward under 4 different scenarios. So basically business as usual, no change, then lowering the emissions, but not having any adaptation, high emissions with adaptation and low emissions with adaptation. And adaptations is not how geneticists use it, but adaptation in the sense of any biological, ecological, or a socio-economic intervention. Okay, that you can basically do to lower the stress on corals besides warming. So the outcome of this is basically twofold. There's really only one trajectory, or there's only one scenario which changes the trajectory, which is lower the emissions, which is here. So this is the non-negotiable part. We need to find a way to lower CO2 emissions, but also that we need this adaptation thing, okay? So we need to understand what active interventions can we do to support corals. And I think this is actually really good news, right? Because me as a biologist, I can work on this biological angle of adaptations. What can we do? And the other important point that came out of this is that we basically have to get our act together. So we have 20 years. It has to happen in our lifetime. So this is why I'm here talking to you. And then I convince 1 to 10 people that you have to do coral biology, and then I basically accomplished my mission. But we just heard that coral reefs are super biodiverse. There's probably millions of species in there. So if you want to start with biological adaptation or active intervention in the biological part, where do you start? And there's a tagline which we use in coral biology, which is called "Save the Coral, Save the Reefs," right? So if you make sure that the corals are there, everything else will follow. And there's one notion that comes from this idea of ocean optimism, which is called bright spots. So bright spots is a very nice concept and is a very loosely defined term. So bright spots can mean a lot of things. Any place where corals are more thermally tolerant, where you have higher coral cover, where you have more fish as expected in comparison to reefs in a similar situation. But these are basically bright spots, are the spots or are the coral reefs that we need to understand. If we understand what makes a bright spot, then we might find a solution. You probably find this picture as pretty as I. This is an actual coral reef in the very far out Great Barrier Reef. So the Great Barrier Reef in Australia, of course, they have it's a very rich nation. They don't have fishing pressure. So this also, diving in that reef, normalized my expectation of how many fish you have to have in a coral reef or you can sustain in a coral reef. This really felt like diving in an aquarium. There were like fish everywhere, and usually fish swim away from you. In this case, everything was swimming towards you, which was also a little bit intimidating. Okay. So how do we find these bright spots? This is just a little exercise. So we have about 1,000 coral species. We have about 1,000 algal species that associate with. This is all estimates. We actually don't know the real number. And then we find about 1,000 different bacteria associated on each coral colony. And we have an unknown number of environments. And so if you do the math and you want to screen this empirical space for bright spot coral holobionts, you end up with a very big number, right? You will never be able to basically screen this. So you have to somehow find a way to constrain your search space. And this is actually the motivation behind what you already heard in the introduction, the CBAS, Coral Bleaching Automated Stress System, which was originally conceived by Dan. And basically we wanted the same thing. It's like Steve Jobs found Steve Wozniak and funded Apple. So we met in 2018. We basically had a vision that somehow experimental screening takes way too long, right? You don't have to run a 3-week experiment to basically define whether a coral does better than another coral. What we had basically envisioned was something like a standardized coral stress test. So if you go to your primary physician, right, you get an electrocardiogram. The primary physician is not a cardiac physiologist, but he can read out this EKG and he can tell you whether it looks normal or not. And this is good enough, because if it looks normal, you're free to go. If it looks weird, then you go to the expert, and then you basically look further what's going on. So this is kind of the philosophy that we had with that. This is how that thing looks like. You can literally pack it into 2 suitcases. So we always had this idea that, corals are underwater. So you have to devise an experimental screening system that you can actually bring to the corals. The minute you have to bring the coral to the lab, unless you live in Hong Kong or in very few other places, it's actually very hard to have direct access to corals. So you can basically pack it in suitcases, and this is set up in a hotel room, and this is set up in an Airbnb. I have set it up everywhere. It works, okay, but I can also tell you it's better to ask for forgiveness after the fact than for permission before. People have usually very little tolerance that you tell them that you run experiments in their living room, even if it's for corals. yeah, so what you can see is basically, this is important later. So these little things are all coral pieces, and we have basically 4 independent tanks where we can basically assess coral performance. And the idea literally is to put corals on a hamster wheel and see how fast they can run. So this is what you can see here. This is the 18-hour test. This is basically a temperature cycling profile. And what we did, I mean, there's different philosophies, right? There's the people that want to run very long aquaria experiments where they collect the coral, then they put them 2 weeks into the aquarium to acclimate and so on. So we scratched all of that. We wanted to have it as quickly as possible. So once we collect the coral, our idea is to get it as quickly as possible into this test. So you're as close as possible to the actual reef condition. And then this is where these 4 tanks come in. So I showed you a colony, which is also nice because you can collect 4 pieces from the same coral colony. So you have the same genotype in 4 different conditions. There's very few animal systems where you can actually do this, right? So you have the same genotype with the same life history. You can expose them to 4 temperature treatments, and then you can pretty much measure its performance. So what they do is basically, or what we do, this is a day in the reef. You wake up as a coral in the morning, then the temperature rises slightly until it's noon, and from noon to 2 o'clock or 3 o'clock, it's very warm, and then it gets a little bit cold, and then you have this overnight recovery. So we basically mimic a day in the reef, with the difference that these different tanks have different heat stress scenarios. Like this is benign, this is a little heat stress, this is a lot heat stress, and this is crazy heat stress. But what you can actually see, we array these corals in bits and pieces on these trays. And after you run this heat stress array, so this is basically 10 different genotypes, right? This is 10 pieces from 10 colonies. At the baseline temperature and at the high temperature, and you can clearly see that they are different, right? So this coral is still colorful and this coral is fully bleached. So this is actually one of the first photos we did when we developed this test. So it's basically a means that you can differentiate genotypes. And this is just because it's fun. Does it play? Oh. Okay, this is a video. How do we collect corals? We bring a drill underwater, and then it's very quick. You can basically drill it. It takes 5 seconds, and then you have this super standardized pieces. It's actually very convenient. Okay, so now you have these 4 temperatures, and then you basically measure performance. What we measure is photosynthetic efficiency, and this is your 4 treatment temperatures. Each dot is a measurement from a coral fragment, which represents a colony. And you can see that the corals from the red population can hold out much longer with increasing temperatures than corals from the blue population. So this was already great, right? And the trick here is that we didn't follow this typical binary experimental design where you have heat stress and control, but we did something that we borrowed from the medical literature, which is called a dose response curve. So this is what people basically do with drugs when they want to see an effect, right? They basically measure effect in terms of concentration. So it's a similar concept here. We basically measure performance over increasing temperature. And what we then thought, also analogous to the LD50, who's familiar with the LD50, lethal dose 50? So the concentration of a drug at which 50% of your experimental animals die. So we have a similar concept, ED50, effective dose 50. So the temperature at which you lose 50% of the initial performance. So what we now have, sorry about that. What we now have is basically a single temperature time point that you can use to compare coral populations, right? And this, of course, makes it very convenient, makes it very standardized to compare coral. So in this case, the blue population has an ED50 of almost 35, and the red population has a much higher ED50 of over 37, right? So if you were basically to decide what is the more thermally resilient coral population, it would be the red one. We have recently expanded this concept. So of course, once you have this dose response curve, you can do all kinds of fancy things. So we now have ED5, which is the temperature at which there's an onset of stress. I just mentioned it here because it's actually very interesting. There's regions in the world where the ED5 is at a temperature that is below the current water temperature in the summer. Okay, so these corals are baseline stressed, even if they are not bleached. So this is very bad news. And ED95 is basically the thermal limit. This is like 5 seconds before you're dead, but it's also important sometimes to compare this. Okay, so having done all of that, as I said, back in the days, I was in KAUST, which gives you access to the sea coast. The Red Sea is actually, it's a pretty narrow, pretty deep ocean with very thermally tolerant coral, but it has a very strong gradient. So it's very hot in the south, it's pretty cold in the north. And we basically brought our test and we measured corals along this temperature gradient. And what we found is actually very interesting. If you correct it for the average summer temperature, all these corals showed more or less the same temperature tolerance. Yeah, this is a very important concept because a lot of times I hear people that Red Sea corals are so thermally tolerant in comparison to Great Barrier corals, but the average summer temperature in the Great Barrier Reef is also much lower. So if you actually compare this, when you deduct the average summer temperatures, all of these corals are more or less the same. They always have the same distance, except for the northern Red Sea. So here they actually peaked and they were way beyond this normalized factor, which is something we already knew intuitively, right? So northern Red Sea corals, this is well known in my world or in the literature. They are amazingly thermally resilient. And the idea is that I just quickly give it to here. So random knowledge. At one point, the Red Sea dried out and then corals had to repopulate. And all the corals that repopulated in the north basically had to go to the south. So you have the temperature tolerance from the southern regime, but you're in the northern Red Sea. So you have a much higher cushion than you actually need. And this is kind of what we verified with this test. So all of this we did in 2 weeks experiment time. So I think this really highlighted the power or the opportunity that you have here. And then, of course, you can plot this and there's a high correlation and so on. And even the New York Times picked up on this because, of course, good news in the coral reef space are like very rare. So it's always nice if you can report on it. But this is kind of what the idea was. So then we took our little test and we brought it in a lot of places. So this is in Guam. You can see that there is no diving required. We literally kneel in these corals and we collected them. So very, very warm, like hot tub warm. Here's the setup. And they showed a similar pattern. So they had this much higher cushion space than corals from other places. So what we basically communicated from this is this vision that you basically bring your little boxes, you travel the world's coral reef and you measure it everywhere. And then based on these bright spots or on these corals that have this exceptionally high thermal tolerance, you can basically develop a conservation map. And then once you know where you find these corals, you can, of course, also look for the environmental conditions of these corals. But not only that, once you have these corals, of course, you can also examine them molecularly to actually understand what they do different to their conspecifics, which are more sensitive. So, of course, this all sounds great now. So Paul G. Allen is the co-founder of Microsoft, besides Bill Gates. So he has a family foundation, and they basically funded us to do this. But again, for all students here, this was 3 years in the making, okay? Like, if I have to tell you one thing you need as a researcher is perseverance and persistence, okay? I called these guys every month for 3 years, and then they invited us over. And then we waited another year, and then they invited us over, and then we got the bugs, okay? So you really have to show perseverance. But basically what this project is about, we went to the major reef regions in the world. So the Pacific, the Caribbean, I did the Arabian Seas and Australia, and we did this standardized approach with molecular examination. But also because this is such a versatile platform, a lot of people actually adopted this approach, right? So this is not that this is the best way to measure temperature tolerance, but it's a very standardized, reproducible way, and a lot of people picked up on this. So I started collecting papers with people actually doing the short-term acute thermal assays, which you can see here. So this is not like in an Apple show where they always show you cumulative sales. So every year you have more sales because you have the number from the previous year plus the actual number. This is total number, okay? This is per year. And this is really started from nothing in 2020 and has a nice uptick. I think there's now 200 or 250 papers on that. What you can also nicely see is the COVID lockdown, right? So there is stuff that is really globally affects us. So I found this interesting. But anyway, so the point is what you can basically build, you have standardized values from a standardized setup. So each on their own, it's one experiment, but because you have many experiments from many different people, for many different time points, for many places in this world, you can actually integrate these data over time and over sites, and then you can get these time integrated observations, which are very valuable. So what we built here is basically what we called Google Maps for Coral Conservation. You can actually go online on coralfuture.org. Again, I mean, the other thing I can tell you about my career is that I talk with a lot of people and I listen a lot. So the guys that I did this with are bioinformaticians from St. Petersburg that I met online while we were discussing something else. But anyways, what you can now do is you can basically, in a very simple manner, you can ask very complex questions, right? You can pretty much ask which is the most thermally tolerant species globally, or where's the most thermally tolerant coral, and so on. And this then gives you, of course, access to, is this common environments? Do you have universal climate metrics and so on? I think we have 250 uploaded experiments right now, but this, I hope this will be growing. And of course, everybody can become a member and upload their data. Like this is really becomes better the more people use it. Okay, so we can skip this. This is, I just became a fan of having a lot of information on one slide, but I told you everything already. Okay, standardized phenotype diagnostics, ED50. Let's talk about active intervention. Again, a lot of this I already showed you, but it's just to mention that we have 10 to 20 years to figure this out. And I think what people don't appreciate enough is there is a lot of coral restoration going on, but we don't have any hard data to actually understand what is how to restore corals, right? Like, because these like long-term success, or you basically restore a coral if you plant it in a reef, if that coral reproduces and build a new coral, and there is no experiment that runs long enough to show this. But pretty much last month, so we have so little time and people have money and they want to invest it to do something good. So this restoration investment is accelerating faster than the evidence to know what to support. This is like an absurd situation we are in, right? But this is a problem at large. So Nature published, or in Nature it was published 4 weeks ago. You can read it, that this conservation at large is an evidence problem. That we actually, a lot of this is how do you say it? It's anticipatory of what's the right thing, but there's very few hard data to support. Again, me and my obsession with standardization. So if you go into the coral reef world, there's a lot of restoration projects which are called a success, but there's no common metric that actually proves this success. So what we need is a standardized resilience metric. And I mean, you probably figured this out already. If you go to this retirement home in The Simpsons, there's a sign on the door, "Thank you for not discussing the outside world." So that's what we cannot afford anymore if you're a coral reef researcher, right? You basically, in my lifetime, I saw these ecosystems going down. So what we built in Indonesia and Gili Asahan is we want to transfer what we learned into the real world and actually make this actionable. And this is a collaboration with Highway. Lou from this university, professor, I will briefly touch on these things that we do there. So thermal pre-screening, you already know the story, but again, I have a lot of pet projects in the world's ocean. So 5 years ago, I just did a sea bass for the fun, and I planted the top and the bottom ones in this reef. So the bottom ones are white, and if you go now, they are tiny, and the top ones are huge and colorful. Okay, so my mom can differentiate, which is the good coral here. Very easy. And then, of course, the big thing, microbiome intervention, right? So again, this is not so much in the common knowledge, I would argue, but this is big in the agricultural world. Okay, so the market is 10.8 billion and it's suspected or projected to be 42.7 billion. So the plant people, agriculture, aquaculture, they have all figured it out. The coral world is late, but this is the trajectory. One of the first studies that actually showed that bacterial community or bacteria might matter was a study we did in 2017, where we basically showed two things. A, that you can transfer microbiomes between heat susceptible and heat tolerant genotypes, and B, crazy enough that the susceptible genotypes become tolerant, the susceptible colonies become tolerant colonies if you transfer the microbiome. So then, of course, this gave this idea of, why don't we use the microbial help to adapt corals to become more tolerant? There was a proof of principle study by Highways and my collaborator Raquel Peixoto. So she basically identified a probiotic consortium. She gave this to corals under heat stress, and there was an increase of 40% survival. So you will maybe not appreciate this yet, but you're very happy about a 2% increase or decrease in your experiment. So this number is crazy. Okay, it's also a nature-based solution because you take the bacteria that are already in the environment. But what needs to be said is that we have no idea how this actually works. And the big bottleneck in every system is long-term association, right? And this is also the cost factor. If you have a microbe, a probiotic bacteria that provides a benefit to the host, applying this is what costs the money. So every time you have to send somebody underwater. So if you apply it once instead of many times, of course, this is good news. So this is at a time when he's sitting there, the man. So again, you really have to appreciate this. This is a universal problem, transient colonization and long-term efficacy of probiotics, okay? And the way people addressed it is that they screened for genes who they inferred to be associated with a benefit to the host. But Highway actually came up with this idea to invert this search pattern, because once you're microbe and you undergo a trajectory to commitment to host, there are specific genomic signatures which you display and which you can identify genomically. So he identified them in a specific bacteria, and sometimes you have to just get lucky. So using our CBAS tests with the inoculate of this bacteria, we could actually prove a thermal tolerance increase. And then they did an experiment in Hong Kong waters where the corals that got this bacteria actually did much better in a natural heat stress event that was unprecedented. Okay, this was the craziest heat stress recorded. You can see the lower things here is what corals usually experience. So the peak really, the curve really peaked. And in the same time, we could also show that these bacteria are tissue associated. Okay, so this is very exciting, right? You have now tools, you have standardized thermal screening, and you have this probiotic search pattern where you can pretty much look for this, bright spot coral. But of course, what I just told you, long-term trajectory. So one good way to assess long-term trajectories, eDNA, I don't want to spend too much time on this, But this is also what we are developing in collaboration with ETH Zurich and Wild in Sync. So I'm also not a fan of duplicating efforts. They do this. What they didn't have is a good eDNA barcode for corals, which we developed. So now we're trying basically to do this. But the idea is to follow this long-term trajectory. In the interest of time, I skip this, but I just want to mention machine learning and AI in general is a huge opportunity to basically annotate underwater pictures, okay? And there's now various things, even so deep reef map, you go in a reef with a GoPro camera, and based on this movie, there is now algorithms that can actually tell you coral cover and coral diversity based on this. So this is a complete game changer. If all you need is an underwater camera to assess benthic cover, then this is very good news. Okay, but this is basically my last part. So we need a resilience measure, right? And a resilience needs to be defined, and we can standardize indicators if we throw everything together. So we have our acute assay data, we have our probiotic intervention data, then based on this automated analysis with eDNA in images and the temperature we get from satellite data anyway. So if there's any professor of mathematics, don't look at this formula or equation. The idea is that you can integrate all these different things, and then you can basically, well, what is integration? You measure the area under the curve, right? If you basically have a trajectory over time, disturbance, recovery, intervention, this is how such an integrated resilience measure would look like. So this is something that I also want to do. But the point is, it's not that resilience is not a claim anymore. It is basically a testable and a falsifiable property. So, which is also what I want to tell people. Success is not that everything works. Success is that you understand why stuff works or why stuff doesn't work. This is success in the scientific field, okay? And if we all do this together in a common way, then we can also tap into cumulative learning. So this is my last slide, just shameless self-promotion. So there is a new Tara Coral expedition coming up in the Coral Triangle. But what you have is a boat, which in my point of view is a standardized laboratory. So you bring a lab to a lot of reefs and you can do a lot of standardized methodology. And this is the type of data you need to actually understand what drives coral resilience. Okay, so with that, I come to my summary, but I don't have to read this to you. But I think, so the situation is dire, but it's not hopeless. And people always ask me if I want to live in the past or the future. I think I would still always choose the future, okay? There's no more exciting time than now. Like, we actually have the scientific toolbox that we can use to tackle these problems, and the path forward is science in a collaborative manner, I think. So really, we want to move from aspirational solutions towards this transparent, science-driven hard data, which can actually support reef persistence. Okay, and with that, I want to finish, and thank you for your attention and for showing up. And yeah, I'm happy to take questions. I don't know if this is part of it or, okay, yeah, I shut up. Thank you.
Let us express our gratitude again to Professor Voolstra for his wonderful lecture. We now move on to the Q&A section. May we invite Professor Lam Hon Ming to come on stage and moderate this section? Professor Lam, please. Please feel free to have any questions to Professor Voolstra in person or via the webinar.
Okay, so it's an excellent talk. So will our students have any questions? Do you want to know something more? Or you have some opinion? Or colleagues? Yes, please.
Thank you, Chris. Excellent work. I'm a practical question for you. for the 2 suitcases that you bring along, what about the water supply? I mean, I saw the water piping, and then you connecting to the Airbnb or the hotel room inlet, and then you just change the salinity or the temperature with the systems.
Yeah, so good question. We collect reef water, so the very water that the corals are in, and what we use is these water cooler jugs, and we just fill them up and bring them to the hotel room, and then this is the reservoir. But again, the idea is to keep everything as native as possible. Yeah, so as I said, don't let the landlord know. It's usually a water party.
Okay, so another question? Yes, please.
Thank you, Professor Voolstra. I'm Mark from the Department of Medicine in CUHK. So it's a great talk. My question for you is, because I'm thinking about evolution. From evolution standpoint, do you think if the global warming is slow enough, the coral reef will develop sort of some resistance to the temperature rise, and then in the future, maybe there will be populating the world again with some temperature-resistant strain of the coral reef?
Yeah, it's a good question. We actually work on this right now. So there's already a lot of selective pruning going on. So if you go to places that had a lot of what we call marine heat waves, so very extreme events, if you go to places, these corals are usually very uniform, which means that they were already selected and everything else died off, or that's one interpretation, right? If you go to other places that were basically spared so far, you usually find a much higher population variability. But closer to your question, corals will always survive, I think, but whether you will have functional coral reefs is the other question. So I think corals can survive, and at one point in the very distant future, we will have coral reefs again. But can we afford as humans to basically let coral reefs die right now? It's first for this ecosystem, but I think it's also this, you're on the highway and you drive by an accident, and then you drive slower for the next 5 minutes, and then you speed again. If we basically accept that we don't have coral reefs anymore, then it's much easier to accept that we have the next ecosystem not anymore. And I think then you become into this domino thing. So I think psychologically, it's a big danger to let this one thing go, because then it becomes also much easier to let the next thing go.
Okay, thank you. Yes, please.
Hey, Chris. Have you seen any evidence of corals shifting their distribution towards rather subtropical or temperate zones due to the increasing temperatures in the tropics?
Yeah, also very good question. And it's very hard to find a good answer to that. So localized, you will actually see this. Like there's a study from Japan, I think, where they can actually see this poleward movement of corals, but globally it doesn't work because of water depths and nutrients and so on. So as I said, the one thing is the tropics and the temperature, but the other thing is the water depths and a lot of other factors. So there's not enough move space, basically.
Maybe I follow up on the evolution question. Right? So when the temperature rises, yes, there are some corals survive, but then it seems like that the biodiversity decrease, right? Because you have the same kinds of coral that adapt to a very specific environment that is high temperature. So in the concept of evolution, biodiversity is a safeguarding the species to survive, right? So if we keep on increasing the temperature and select only as a subset of the coral, will it also bring endanger the long-term survival of coral?
Yeah, I mean, that's exactly the narrative, right? Like what I showed you, or I think that a lot of people would actually say that. So the objective is to conserve enough genetic diversity so coral reefs can be rebuilt once we reached basically carbon emission or carbon zero neutrality, I think, right? So it's exactly what you said, that the big danger is that you lose so much diversity that you end up in a situation of some wildlife where the population size is so small that you get all these bottleneck effects that you wouldn't have in bigger populations. And then it's very hard to get out of that.
Okay, thank you. So any more questions from the students? From the top there or at the floor? Okay, yes, please.
Thank you for your talk. I wanted to find about your perspective about current coral restoration strategies. Do you think they would be useful, or is there any way we can better target our efforts towards that?
Can you repeat the last part? Sorry.
Yeah. Do you think current coral restoration efforts are useful, and is there any way we can better target our efforts?
Yeah. Oh God. We amongst scientists, so we have to criticize. So. How to say, so the minute you step out of is what I said, a lot of people are not aware that coral reefs exist or about their ecological or economic importance, right? So I think restoration, even if it doesn't serve anything else, it serves the purpose to make people aware of the problem. But then, as I also showed with this Nature article, we have a problem at large that we are suddenly in a situation where we have to restore things and we don't actually know how this works. But this notion that if you put the same coral underwater that bleached before, it will bleach again, it really amounts to the reproducibility of a scientific experiment. So you have to add a factor X, and probiotics would be one way that would be this factor X, I think. So I think there's a lot of restoration projects, also some of them at scale, but somehow there's not a lot of overlap between researchers and restoration people or practitioners, as it's called. So I think we need to reach across the aisle, but this is basically Highway and I got money to do a project in Indonesia which tries to exactly accomplish that. You try, or you generate the scientific evidence while you do it, right? And I think this is the right way. Like, we're running out of time. You cannot wait for the perfect solution. If you have a good understanding what works, but you still need to apply the scientific method. It cannot be purely aspirational. So I think, again, it's right. It's a longer answer, and nowadays the world is black and white, but details matter is basically my two-word answer.
Okay, any more questions? Yes, at the back. Yeah, there's a student at the back. Yeah, cool. Yeah, please stand up so that they can see you.
So I'm from the Department of Engineering, but on a slightly less engineering-related note, I want to ask, since you scientists, you focus on finding the specific, by doing the scientific method, you figure out what might work and what might not work. But then you also mentioned in your speech just now that public opinion, like human minds, they respond slowly to what we might find out in the sciences. So a lot of these slow effects, they're reducing the coral reefs and their negative effects. What kind of steps do you think we can break this barrier between what we can know from science and getting people to realize that these big problems are happening slower than the human time frame can realize?
Yeah, very good question, I think. So traditionally, scientists live in the ivory tower, right? And they are perfectly happy with their experiments. I think this is something we need to break, that we have to occupy a bigger talking space, so to say, in the public and in the public discussion. Like, because if we don't speak up as scientists, somebody else will on your behalf. And I mean, especially nowadays, like, again, also courtesy of COVID, I think partly the scientific method is not in a good light, right? And somehow for humans, it's intricately easier to focus on the negatives than on the positive, right? We are never happy. But one lesson out of this COVID thing, we developed an mRNA vaccine in the time course of a year. I mean, how freaking amazing is that? And how few people talk about this. I still find this crazy how much we can accomplish in such a short time. So I think what you cannot forget, what I said before, it's dire, but it's not hopeless, right? Like, humans are pretty amazing at doing stuff. And like in small interactions, everybody is well intended. Somehow at bigger gatherings, things fall apart. And I think having more people contribute to the discussion will already help. And like also getting people excited about science and research. I think there's really, science is the future, or there is no future without science.
Okay, there's a question from the internet. So coral restoration use microbiomes is a very insightful approach. I would like to ask, what are the main challenges associated with this method? Could there be potential concerns about reducing microbiome diversity within coral reefs and possibly in the surrounding environment as well?
Yeah, also good question. Okay, so first we have to acknowledge that there is no undisturbed nature anymore, right? I was in a submarine at 4,000 meter depth in the ocean, and the first thing I saw, a plastic garden chair. Like, how is this possible? But there is no place where you have this undisturbed, pristine nature. Everything has already an anthropogenic impact to it. I think this is the first thing. So it's not that we basically break the porcelain, right? We are already very well in the process. The other thing is that, of course, it depends, right? So the probiotic approach that we follow is you actually identify bacteria that are present in the very environment as a solution. So this is not, I'm not advertising genetic engineering of bacteria. So what you're basically changing is you're changing the concentration of bacteria that are already present. But this, of course, we also have to understand, and this is what we're testing. But basically, it's not that we're in a situation where you do it and there could be a detrimental effect, or you don't do it and then everything is great. If we don't do it, it will die anyways, right? This is okay. This is a little bit extreme, but it's not going to go away on its own. So I think we have to get used to this idea that we have to actively intervene. And if we do this, of course, we have to understand if there is side effects and off-target effects and whatnot. But it's not that if we don't do it, we live in a perfectly happy world. So, sorry, not doing anything also carries a risk. It's basically the short answer.
Yes. Any more questions? Oh, maybe I do a short summary here. Professor Voolstra posed an interesting question. Do you want to live in the past, or do you want to live in the future? So if you choose to live in the past, you will feel hopeless, right? So if you choose to live in the future, you need to take action. Solution may not come with a perfect solution, but something must be done to alleviate the situation. And in particular, Christian has said, people are willing to spend resources in coral restorations, but the resources must be put in the right hands. Therefore, scientists must speed up and educate the public what is the best way to go forward. And we should be brave enough to take actions, and that will contribute to our future. I hope that this is also something that our students can learn tonight from Professor Voolstra. Okay, with this, let's give Professor another big round of hands, and thank you for the great and excellent presentation.
Once again, thank you, Professor Voolstra and Professor Lam. May we now invite our acting head, Professor Lee Ho-man, to come on the stage and present the souvenir to Professor Voolstra and have a photo taking on the stage. Now we sincerely invite Professor Lee and Professor Voolstra to leave the hall first. Thank you. To gather the feedback on the lecture, we would like to invite both participants attending the Zoom webinar and in person to complete a questionnaire. Now, may we first invite our guests to leave the hall now. Our college students, please remain seated. 其他同學請安坐，相週會繼續進行。